Hello everyone. I welcome you all to Biology Lessons with Kalyani, and in today's class we will be discussing about the part 2 of cell cycle and cell division, wherein we are going to discuss the meiosis, which is a second type of cell division. Let's first look at what are the different topics we are going to see today. So first we will see why we need meiosis. Second, we will be studying meiosis part 1, and then we will be looking at meiosis part 2. Then we will discuss what are the number of chromosomes and DNA content in different cells undergoing meiosis. Finally, we will be talking about the biological significance of meiosis. Now, if we see in case of organisms that reproduce by sexual reproduction, now we get one of the gamete from the male parent and another gamete we get it from the female parent. So in that case, how do we restore the chromosome number, right? And we know that for every species the number of chromosomes is always same or constant, and that cannot be changed. So the chromosome number, the only way in which we can maintain it is by production of haploid gamete, so that the haploid gametes can fuse together and form a diploid zygote. To make it more clear, so let us consider that we have an organism which has the chromosome number of 2n. So and we get one set of chromosome from the male parent and another set of chromosome from the female parent. So how do we combine them? And both of them combine together, and it should have a fixed chromosome number, that is 2n. So only way is that we have haploid gametes from the male and haploid gametes from the female. So in this case, the meiotic cell division comes to the picture, which is basically the reductional division. So meiosis is a specialized cell division which takes place in the diploid cells, and from a 2n diploid cell, we get 4 gametes which have n number of chromosomes, right? This specialized kind of cell division reduces the chromosome number by half, resulting in the production of haploid daughter cells, and we call it meiosis. Now, meiosis ensures the production of haploid phase in the life cycle, so that after fertilization, the chromosome number or the diploid phase can be restored. Is that clear? Now, if we compare this meiosis, the stages of it as that of the mitosis, it is meiosis stages are a little more complex. Now, meiosis typically has meiosis part 1 and meiosis part 2. Both meiosis part 1 and part 2, they have their own prophase, metaphase, anaphase, and telophase. And meiosis and meiosis part 1 and meiosis part 2, in between them there is a short gap phase, we call it interkinesis. But in this gap phase or the short phase, which we call interkinesis, there is just protein and RNA synthesis. It is to be noted that no DNA synthesis take place. So if it is asked that what would be the sequence of like the different stages in case of meiosis, it will be interphase, which will have G1s G2 phase, followed by meiosis 1, then interkinesis, and followed by M2. So there will be no S phase here, right? It's a very important concept. Now, meiosis part 1, if we go, it will be further divided into prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis, the prophase 1 of meiosis part 1 is a very long phase, and it is further divided into 5 substages. And the name of the 5 substages are LZPDD. Okay, so you can remember it by LZPDD. D. So it refers to L refers to liptotin, zygotin, pakitin, diplotin and diakinesis. So let's look into the different stages in detail now. So this is the liptotin stage, which is the first stage of meiosis part 1 or the prophase 1. Now we are covering here prophase 1. The first stage of prophase 1 is liptotin stage. So basically in interphase we all know that the chromosomes, the content of it is doubled. That is, the sister chromatids are synthesized. In addition to that, the cell organelles are formed. Right, so we have already discussed that in part 1 of our lecture, but the chromatin fibers, they remain in a very tangled up way, or they are present as thin threads. So first thing which we need for a division is that we need compaction of the chromosomes. So the chromosomes condense and become gradually visible in leptotin stage of the prophase. So you can also visualize them under the light microscope in this leptotin stage, but still the chromosomes are not very much condensed like that we get in the metaphase stage. So they look as a they look at the bouquet. Okay, so leptotin stage is also called as the bouquet stage. So this is how the chromosomes in case of leptotin look. It is to be noted that in this stage nuclear envelope is still intact. The black color is from the chromosomes from the male and the orange represents the chromosome from the female parent. So chromosomes become gradually visible in leptotin stage. Compaction of the chromosomes still continues. Okay. Next we go to the zygotin stage. Now in zygotin stage, the nuclear envelope is still there. Further compaction of the chromosome or condensation of the chromosome still takes place. And in this stage, what exactly happens? See, the similar types of maternal and paternal chromosome, which we call as homologous pair, they tend to combine with each other, or they are paired together. Pairing of the homologous chromosomes takes place in the zygotin stage. Is that clear? So what happens? What are these homologous chromosomes? Homologous chromosomes are the chromosomes from both male and female parent that resemble each other in the structure, shape, number and pattern of the genes present. So they pair up together in the zygotin stage. So as they pair up, the chromosomes are present on the top of each other, not side by side as it is looking here, but they are actually present on the top of each other. I have a diagram for it which I will be showing later, but for now remember that when the two homologous pair come together, they are bound by a complex called as a synaptonemal complex, and this complex you cannot visualize under the light microscope. You need the electron microscope for the same. Now this process of pairing of the chromosomes is called as synapsis. Synapsis takes place by the formation of the synaptonemal complex. Is that clear? Another important thing which we need to remember is that when you look at the chromosome which we obtain from only one chromosome from the male parent, let's say, if you observe it, then it is having two sister chromatids because it is duplicated in the S phase. And because it has two sister chromatids, we call it a dyad. But as this chromosome is not paired up, we call it univalent chromosome. So this univalent chromosome, where do you get? You get it in liptotin stage. But as we proceed to zygotin stage, the homologous chromosomes are paired together. So this pairing brings the two paternal and maternal chromosomes together, and together, because we have four chromatids in them, this structure is called as tetrad, And also, as there are, you know, two homologous pair, it is called as bivalent. So bivalent chromosome is nothing but a pair of homologous chromosome. So the formation of the bivalent chromosome and the formation of the tetrad, it begins in the zygotine stage, but we cannot clearly demarcate the tetrad, that structure we cannot see it clearly because they are closely associated together by the synaptonymal complex. But the formation of the tetrad, formation of the bivalent, it takes place in zygotine, right? Another thing you need to also remember here is that when you consider a chromosome, the chromatids present in the same chromosome, it is called as the sister chromatid. Okay, this is the arm of the chromosome. This is one chromatid, this is another chromatid. Both the chromatids are present in single chromosome, so we call it sister chromatid. But if you consider the chromatids of a homologous pair, or maternal chromatid of a maternal chromosome and a paternal chromosome, both are called as the non-sister chromatids, right? I hope that's clear. Let's move on to the next stage, that is packeting. So in packeting, what happens? The four chromatids of each bivalent chromosomes become distinct and clearly appear as tetrads. So formation of tetrad, it takes place in your zygotine stage, but it becomes visible in the packeting stage. Okay, see, this is how the packeting, the chromosomes will look like. The tetrads are clearly visible. That means that the the chromatid arms, the four chromatid arms, we can clearly see them, right? There is formation of this packeting stage is very important. In this stage, recombination nodules are formed, which are basically sites at which crossing over takes place between non-sister chromatids, right? We know that meiosis just not leads to the formation of gametes, it is also responsible for the genetic variability. Now, how does this genetic variability come from? This genetic variability comes from the crossing over between the non-sister chromatids. Let's say this is your maternal chromosome and this is your paternal chromosome. Okay, let's demarcate it. This is your maternal chromosome and this is your paternal chromosome. Now what happens in case of packeting? There is crossing over. First the two will come together in case of your the zygotine phase. After that, the two chromatids, they will cross, they will exchange the genes in between them. This exchange of genes is called as the recombination, okay, or the crossing over. Recombination or the crossing over, or simply crossover. So crossing over takes place in packeting. First they will come together and in the zygotine they will lie very close to each other. Let me make another one. They will lie very close to each other, uh, you know, and they will be joined together by the synaptonymal complex. This is called as this is the process is called as synapsis. This is obtained in zygotine, and in packeting what happens, the actual crossover takes place. Look, now this diagram is showing the crossover or the exchange of genetic information or genes between the non-sister chromatids, that is between the chromatids of paternal chromosome and maternal chromosome. This crossover takes place with the formation of recombination nodule, and the enzyme recombinase is involved in crossing over. It's very important thing. So where is recombination nodule found? Recombination nodule is found in packeting. Okay, whereas tetrads clearly visible, tetrads are clearly visible in your packeting stage. Okay, and where does crossing over take place? Crossing over also takes place in packeting. Okay, but it is important to note here is that the synaptonemal complex, which is responsible for joining together of the two homologous chromosome, that is still present here, that is not completely dissolved. Okay, now recombination nodule, as you can see, this is the site where the recombination is taking place. This is here is your recombination is taking place. Here also recombination is taking place. So both of them are called as a recombination nodules. I hope packetin is clear. Let's now see what is happening in diplotin. The beginning of diplotin is recognized by the dissolution of synaptonemal complex. So the synaptonemal complex, which was holding the two homologous pair together, that was marked in blue. You can see that is just not present here. So as a result of which there is an X-shaped structure which can be seen here. See, this X-shaped structure is called as chiasmata. What are the chiasmata? Chiasmata is the site where the recombination has taken place, right? So sometimes what happens in oocytes of some vertebrates, the diplotin can last up to some months or years. The diplotin is very prolonged, and such cases the chromosomes lie in this X-shaped structures, and that those oocytes are called to be in the dictutin stage, right? So important characteristic of diplotin: dissolution of synaptonemal complex, formation of the appearance of the X-shaped chiasmata. So these important terms are always asked in either match the following or statement based answer. So this is very important and you need to remember it. So you should note here that until diplotin stage we still have the nuclear membrane. Next, let's see diakinesis. So it is the final stage of prophase 1, and we know by the end of prophase 1, that is dissolution of the nuclear membrane. So see, so here what happens, diakinesis is marked by the terminalization of chiasmata. Chiasmata was the site where the recombination or the crossing over took place, and in this case of diakinesis, in this stage of diakinesis, what happens, that chiasmata is that is almost dissolved. We don't need any more recombination, just we can see terminal chiasmata formation, only the ends look to be joined. Okay, so terminal chiasmata is a very important characteristic of diakinesis. Rest what happens here, chromosomes are very much condensed here. There is disappearance of nuclear membrane, nucleolus and endoplasmic reticulum. So with this, with the step of diakinesis, the prophase 1 is like over. So let's see what happens in metaphase 1. So just quick overview about prophase 1. What did we get in diakinesis? No nuclear membrane is present, nucleolus absent, endoplasmic reticulum absent. What about the your chromosomes? The paired chromosomes are having just the terminal chiasmata or are just joined along the ends, right? So what should happen here in metaphase 1? The asters also come are made in your prophase 1. The asters will radiate the spindle fibers. So what will happen, the spindle fibers, they will be attached to the centromeres of the homologous chromosome, and the homologous chromosomes, they will arrange themselves along the metaphasic plate. And you can note here that we are getting one set or one imaginary line or one plane in which the one pair of homologous chromosomes are arranged, and another metaphasic plate or another imaginary line along which another set of the metaphasic chromosomes are aligned. So how many metaphasic plates are we getting in case of metaphase 1? We are getting two metaphasic plates, right? And the each chromosome, how many spindle fibers it is attached to? Each chromosome is just attached to a single spindle fiber. In case of mitosis, we know that a single chromosome centromere is attached to two spindle fiber. But here in metaphase 1, the centromere of a chromosome is attached to just one spindle fiber. So how many spindle fibers we will get? So if we have 46 chromosomes, so 23 pairs will be attached to 23 spindle fibers, on which will be on one side of plate, and another 23 pair will be attached to another 23 pairs of 23 spindle fibers. So total 46 spindle fibers will be present, right? Because we have humans, they have 46 chromosome, 23 pairs of chromosome, and we are separating the chromosome pairs in case of meiosis, right? Because we are getting the haploid chromosomes in case of meiosis part 1. So we will have a total of 23 into 2, 46 spindle fibers, right? Not like mitosis, wherein we used to get your 92 spindle fibers. Is that clear? So let's read out what happens in metaphase. The bivalent chromosomes. Why bivalent? I already told, the homologous pair is called as a bivalent. The bivalent chromosomes align on equatorial plates. See, this is one bivalent, this is another bivalent. So the bivalent pair, they align along the equatorial plate. The microtubules from the opposite poles of spindle attach to the kinetochore of the homologous chromosome. So the microtubule is attaching to the kinetochore of the homologous chromosomes. Next, let's see anaphase 1. So what is happening in anaphase 1? The homologous chromosomes separate. Okay, the homologous chromosomes move towards the opposite poles, with the centromere trailing first, followed by the chromatid, followed by the chromosome arms. Right, sister chromatids remain associated at their centromeres. Right. So in telophase 1, the nuclear membrane and nucleolus reappear and cytokinesis follows. So see, in anaphase 1, essentially what are separated? The homologous chromosomes are separated, and the nuclear membrane is formed around the separated homologous pair in case of your telophase 1. Telophase 1 is just the opposite of prophase 1. So nucleolus will also reappear, nuclear membrane will reappear, right? So what are getting separated here? So what we are essentially getting is one out of the pair of homologous chromosome. So we are getting the dyad. Here your the bivalent or the tetrads were aligning along the metaphasic plate. The result of your meiosis part 1 is the separation of the separation of the tetrad, leading to the formation of the dyad, right? So because each chromosome is having two chromatids here, we are calling it as the dyad, and as it is just one out of the pair of the your homologous chromosome, it is called as univalent, right? So after the telophase 1, if we will have an interkinesis stage, which is the stage between two meiotic divisions. It is generally short-lived. There is no replication of DNA during this stage. So unlike S phase, where the DNA is duplicated, in this middle phase between the meiosis part 1 and part 2, the interkinesis stage does not have any S phase. The chromosomes do undergo some dispersion. That means there is some amount of decondensation, but the amount of decondensation is not that much, right, like that of the interphase. So let's now see what is happening in the meiosis part 2. So, in case of prophase 2, meiosis part 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. Now, this prophase 2 resembles as that of the normal mitosis. The nuclear membrane disappears and the chromosomes become compact. So, essentially, what's happening in prophase 2 is that the whatever haploid number of chromosomes we have got, those chromosomes, they had little bit undergone decondensation in the interkinesis stage. They again start to condense or tend to become thick, because we are again going to divide them, so we need them to be thick, right? So this the nuclear envelope also disappears and the chromosomes become compact. This is followed by metaphase 2. Metaphase 2 is also similar to your mitotic metaphase. So what essentially happens here, whatever chromosomes we have, they will align along the equator. Chromosomes align at the equator and the microtubules from the opposite poles of the spindle fiber get attached to the kinetochore of sister chromatids. So if this is your the aster, the spindle fiber radiating from aster, it is attached to the kinetochore. Each chromosome centromere is attached by how many spindle fibers here? 2 spindle fibers, right? But how many spindle fibers we will get in metaphase 2? Because we got out of the 46 only 23 chromosomes in the cell starting prophase 2, we will get how many? 23 plus 23, we will get 40 46 spindle fibers in metaphase 2, right? Now, how is it remaining same as that of the metaphase 1? Because here a single centromere is attached to how many spindle fiber? Single centromere is attached to 2 centromeres. So 23 chromosomes are there. 23 will be attached to 2 spindle fiber. 23 multiplied by 2 makes it 46. So 46 spindle fibers will be present, right? So that's all about metaphase 2. What is happening in anaphase 2? Anaphase 2 begins with the simultaneous splitting of the centromere of each chromosome, allowing them to move towards the opposite poles of the cell by shortening of the microtubules attached to the kinetochore. So this is basically similar to that of your mitotic anaphase. So centromere splits, as a result of which the spindle fibers contract and lead to the movement of the sister chromatids towards the opposite pole, right? So see, the centromere trailing forward followed by the chromatid arms, right? This is anaphase 2. So the final stage is your telophase 2. That's the final stage of karyokinesis in case of meiosis 2. What happens here is similar to that of your mitotic telophase. Two groups of chromosome once again get enclosed by nuclear envelope. Nuclear envelope reappears. Then cytokinesis follows, resulting in the formation of tetrad of cells. So how many cells are formed here? See, 4 cells are being resulted. This is one cell which was produced by meiosis 2. This is the second cell. Sorry, this is the first cell which was produced in meiosis 1. This is the second cell which was produced in meiosis 2. Both had 23 chromosomes, right? The chromatids of the 23 chromosomes, they got separated here, as a result of which from each cell we are getting 2, making a total of 4 cells, and each cell is having 23 single threaded chromosome. That is, we are getting a tetrad of cells which is having haploid number of DNA, right? The how many like we can the chromosome number will be how much for it? N, and the chromosome content will be also be half, right? We will be looking into the chromosome content and the the DNA content later, but for now you must be clear about from two cells which were produced in the meiosis 1, 4 haploid cells are produced. Because 4 haploid cells are produced, it is called as tetrad, and the chromosome number of it would be N. So these are some of the extra diagrams, not in your it's not in your textbook, but you can just refer it as just for the understanding purpose. So see, this is how the homologous chromosomes are getting paired up, forming divalent. Single chromosome is called as univalent. They are coming together, forming synapses, and after that, crossing over is taking place. This X-shaped structure is called as chiasmata. Right now, this chiasmata is clearly visible in which stage? In your leptotin, condensation of chromosomes while taking place. In zygotin, synapses took place. Then in pachytin, your the recombination took place, and after the synaptonemal complex was dissolved in the diplotin stage, the chiasmata was to be seen, and only the terminal chiasmata were seen in your diakinesis stage. Right, so this is how, if you are look under the electron microscope, this is how the these are the chromatin fibers, this is the synaptonemal complex. Right, see, the two chromosomes during synapsis, they are actually on the top of each other, and they form a recombination nodule, and exchange of genetic information takes place by the process by the help of the recombinase enzyme. So if you see, in leptotin stage, synaptonemal complex is not there. It starts with zygotin. You can see the synaptonemal complex in pachytin also. In diplotin also, it is partially dissolved, and it is completely dissolved in diakinesis. Right, these are the diagrams for your extra reference, just for understanding purpose. So let us now look at the number of chromosome and the DNA content which we are getting in meiosis 1 and meiosis 2. Now see, when we started the meiosis 1, in the prophase 1 stage, the number of chromosome is 2n, and let's say the DNA content is 4c. Why? Because in S stage, S phase, already the chromosome is having two sister chromatids, that is the DNA content has duplicated in interphase. So I will just write it here, because in S phase of interphase, the DNA content has duplicated. That is, first you had started with 1 and it has synthesized the sister chromatid. So if it was 2c, now the DNA content is represented as 4c. Right, so this is your DNA content. Next, what happens in metaphase 1? There is no, if you see prophase 1, the number of chromosomes number is not changed. Okay, just the thickening of the chromosome take place. See, that just the thickening of the chromosome. So the DNA content also remains unchanged. Next, see metaphase 1. When you see metaphase 1, the homologous pair is just arranged along the equatorial line. So we are not doing anything with the chromosome. So both the DNA content and the chromosome number remains same. In anaphase also, what happens? They are moving towards the opposite pole. What is moving? The homologous chromosomes are getting separated, and they are moving towards the opposite pole. Though the reductional division starts here, but because cytokinesis is not formed, so the overall, if you see in anaphase also, the number of chromosomes is same, because we are having each of the pair of the homologous chromosomes, both are there. So both the DNA content and the number of chromosomes remains unchanged. But if we see in telophase, after the telophase 1 is completed, the two cells are formed. So each cell, it is having one half of the homologous chromosome. Another half is in the second cell which is formed. So how many number of chromosomes will be there? N, and each of the chromosome which is having or two chromatids. So what will be the DNA content? Because we are having half of the chromosome, and each chromosome is having half of the the each chromosome is having the two chromatids, two sister chromatids in them, but the DNA content will be still 2C, because there are just, instead of 46, let's say we started with human chromosome 46 chromosome we started, here we are getting 23 chromosomes, right? So 23 chromosomes if we are starting, so the DNA content, if we see 23, 20, we are just having instead of 46 we are having 23, so the DNA content 23 chromosomes, so the DNA content will be also halved here, right? It will become 2C, because half of the chromosome is going to the second cell, right? So let's see what is happening in the prophase 2. If we see prophase 2, now we just thickening of the chromosome, so the number of chromosome and the DNA content remains unchanged until your metaphase 2. If you see the anaphase 2, it is compared to that of the mitotic cell division. So see here what happens, the sister chromatids, they are getting split at the centromere. So even though we started with a number of chromosomes n, actually the two pairs of sister chromatids are separated in case of anaphase, and because cytokinesis has not taken place as a whole, if we are seeing how many number of chromosomes we are getting, we are getting here 2n number of chromosomes, but the DNA content will still remain same, that is 2C. So when further telophase takes place, we are getting one of the sister chromatid, right, from each homologous pair. So this is one homologous pair, this is one homologous pair, two chromatids separated from this, and two chromatids are separated from the other pair. So if we see the how many number of chromosomes we are getting here, we are getting here n chromosomes, and the DNA content, if we are saying, we are having now 23 chromosomes, and each is having just a single chromatin thread. So how much is the DNA content? DNA content is also divided by it is also divided by 2. It is becoming C, because out of the two sister chromatids we are getting one sister chromatid here. So when we started with 4C DNA content, the DNA content is half, and the number of chromosomes is halved after the end of telophase 2, and the DNA content is 1/4 to that of the initial DNA content. This is a very important concept. A lot of questions come from this. I hope that's clear for you. So to summarize meiosis, we can remember that meiosis, if we look at the stages of it, it has one interphase followed by M1 interkinesis and M2. It is to be noted that interphase has only single S phase. Interkinesis does not have any S phase, or DNA is replicated in a single cycle in case of meiosis. Very, very important point. Okay. Next thing is that meiosis involves pairing of homologous chromosome and recombination between the non-sister chromatids. Now, result of meiosis is that 4 haploid cells are produced at the end. Now these are the different stages of meiosis. Important characteristics of prophase stages: lipoptotin, chromosome condensation starts here and get visible for the first time. Zygote, synapsis takes place, a synaptonemal complex, and pairing of the homologous chromosome takes place in zygote. Tetrad formation initiates in zygote but is observed in case of your packaging. Very, very important concept. Just pay attention what is asked in the question. Is it asked tetrad formation starts in which stage, or tetrads are observed in which stage? So if it is asked tetrads are observed in which stage, answer would be packaging. But tetrad formation, which stage it is started, the answer would be zygote. Okay, where is recombination nodule formed? In packaging. Crossing over also takes place in packaging. Which enzyme helps in meiosis part 1? Answer would be recombinase, right? Then diplotin characteristic is that synaptonemal complex is dissolved. X-shaped structure or chiasmata formed in diplotin. And diakinesis, terminal chiasmata is formed. The chromosomes are held together only at the ends. Okay, so if reduction of chromosome, where does it take place? If it is asked, the answer would be anaphase 1, because in anaphase 1 we are actually getting after the anaphase 1 we are getting two cells with only one pair of the homologous chromosomes. So actual reduction of chromosomes take place in anaphase 1. Okay, coming to significance of meiosis. So because of this meiosis, there is conservation of specific chromosome number of species, right? Because the chromosome number is reduced by half in the formation of zygote, the haploid gametes can fuse together, and hence the chromosome number can be restored. Now this, because a crossing over or recombination is taking place, this is the result. This results in variation. So variation in species arise because of the meiosis, right? So that's all about meiosis and meiosis stages. I hope you're very clear about the different stages of meiosis, as well as what happens to the chromosome and how the DNA content is getting affected in the different stages of meiosis. It's a very interesting topic, and I hope you enjoyed understanding it. If you liked the session, please hit the like button, share and subscribe to my channel. And if you have any queries, please feel free to comment below. I will be very happy to answer the queries. Thank you everyone. Have a very good day.